Welcome, everyone, to the Association of Women Endoscopists special Q&A on artificial intelligence in endoscopy, digesting the future, how AI reshaping gastroenterology. I'm so delighted today to have one of my old friends from my days in Boston, Dr. Michael Wallace. He's joining us, and his former fellow, Mariam Al-Hamad. Dr. Michael Wallace is the John C. Anderson Professor of Medicine at the Division of Gastroenterology and Hepatology at the Mayo Clinic, Jacksonville, Florida. He's the former editor of Gastrointestinal Endoscopy, former chief of the Division of GI and Hepatology at, I'm going to have trouble with this one, sorry, Sheikh Shagbot Medical City in Abu Dhabi. And his fellow Mariam was a star, and now she's associate director for IBD research in Abu Dhabi, and she was one of his premier GI fellows, and I think recently hired last year. And welcome, both of you. So delighted to have you as part of our course this year for AWE 2024. I also have my co-founders of the Association of Women Endoscopists on, Dr. Sonia Riker from Harbor UCLA and Dr. Phoenix Nguyen at Hoag Hospital. And we're just going to continue on. So I'm so delighted to have you. AI is such an amazing emerging technology within medicine in the world right now. It's the hottest topic in the world. And I just really want to learn, we all want to learn more about AI in gastroenterology in practice, and many of us are familiar with it being used as a tool for colonoscopy, and I know you've published in it, and people are using this for detection, diagnosis, characterization, and maybe even helping us guide therapeutics. So I thought maybe we'd start there with kind of the premier use of AI that many of us are already familiar, and hoping that you can explain how AI technologies, like for instance, GI Genius, are currently being integrated into clinical GI practice.
Yeah, Marilee, thank you so much. It's a real pleasure to work with you again on this project. As you said, we started both of our careers 25 some years ago in Boston together. It's nice to continue working together. And Mariam, feel free to jump in wherever you want. I thought maybe I'd give just a little bit of a quick overview of the talk that we've recently presented and kind of where AI stands in the GI world. Obviously, AI is having huge influences on society and in medicine as well. I mean, GI is in a fortunate role. I think right now there's 3 subspecialties that are probably most directly impacted today by AI technology. One is radiology, another is pathology, and the third is gastroenterology. And the 3 of these share a common theme, which is we look at images a lot. And so really the first major applications that got through clinical trials in the Food and Drug Administration approval process were these 3. And everybody, if you talk to your radiologist, they're probably using AI systems to help them read mammograms or CAT scans. If you talk to your pathologist, they're probably using AI systems to help them read common pathology, for example, Pap smears and other very routinely performed pathology. And now we're using it in GI. And colon polyps was the first low-hanging fruit. It's what we do most of us every single day is do colonoscopies and to try to find polyps. And there was a clear need to help us find polyps better and improve our adenoma detection rate. But that is just the first app. I often use the analogy of when you first got your smartphone, you might have had a few apps on your smartphone. Now you probably have 50 or 100 at least. And there's thousands, if not hundreds of thousands of apps out there. And I think the world of AI and endoscopy and the world of AI and medicine in general is going to go to the point where you're just running all these apps in the background and you don't even realize that you're using AI. Probably you've seen a little bit on your Microsoft Office tools. You're starting to see these Copilot logos on there, and it's auto-filling text for you and maybe giving you spontaneous responses. But in GI, we're able to use this right now. It was based on the two randomized trials that we presented in our talk earlier. But one of the things that we've talked a little bit about in our recent talk in a project that Dr. Al-Ahmad led was, does it really work in practice? It's one thing if it works in the big trials where there's a very controlled setting, but we've had a little bit of back and forth on, whether it works in practice. You probably all saw the paper out of Dr. Yuri Ladabaum's study out of Stanford that said it didn't really work so well when Stanford implemented this. But we did this project in Abu Dhabi where almost identical design. We implemented it in one of 4 rooms and just monitored our adenoma detection and adenomas per colonoscopy when colons were somewhat pseudo-randomized to that site or to that procedure room, and compared it to ones that didn't. And Mariam, do you want to expand a little bit on the, does this work in practice? Should gastroenterologists be considering this?
So our study showed that the results were almost the same as the one published in randomized trials in USA and Europe, showing the benefit of AI in adenoma detection rate and ADR as well. We implement this project on a lower screening age. Here we start screening at age of 40. So we had a significant results based on a significant p-value in ADR and APC. So we think that it is generalized to probably the Middle East as well. And we think that it should be used. But as Dr. Wallace said, there is a conflicting results have been published recently that showed no benefit of AI on APC and ADR as well.
One of the things that strikes me a little bit about what's the debate right now with, does the colon polyp application work? In a lot of things we do in GI and endoscopy, we often criticize, well, these uncontrolled studies or these non-randomized trials were positive, but we really need randomized trials. Now we have kind of the reverse, right? We have 2 very large multi-center, multinational randomized controlled trials that are positive, and we have mixed results on the non-randomized single center studies, some positive, some negative. So I as a former editor, I always fall back and say, what's the level of evidence? We have essentially, level 1 evidence, 2 large randomized controlled trials that this work. And the other stuff is, is less rigorous. So to me, if I had to choose between which data do I believe the most, I still am going to believe the randomized controlled trials more than the less well-controlled trials.
Yeah, I feel that, definitely these large randomized trials have shown its advantage for adenoma detection rate. But I also wonder what your thoughts are in respect to teaching and education, especially as these large endoscopic companies start integrating it within their endoscopic equipment, what your thoughts on that are for training. And then also, Dr. Wallace, you asked me to write an editorial a couple years ago for GIE, and I did make a little brief comment about AI and its use for polyp, not only detection, but also assisting us with our therapeutics. How do we remove the polyp, and can it assist us to take the entire polyp out? So any comments appreciated on that?
Yeah, I think, I view AI a little bit like someone looking over your shoulder. All of us, when we were fellows, we had somebody looking over our shoulder, and if we didn't see a polyp, our attending physician said, there was a polyp you might have just missed. Why don't you go back? This is essentially a second set of eyes. I think in the end, most of the polyps we find when we're using these technologies, we see it at the same time that the AI technology sees it, and we don't necessarily need the support of the AI technology. But there certainly are some, and I've had this myself, and I've been doing this a long time. It sees polyps that I don't initially see. They might be a little tiny polyp that just gets flagged, but sometimes the really flat, subtle ones get flagged in a way that I tell myself in my head, I tell the nurse in the room, I would have missed that polyp if I didn't have the technology. But we haven't really studied the learning impact of it. I think back though a little bit to, you probably all remember when narrowband imaging first came out. And the early trials suggested that when we first started using NBI, it seemed to help us a little bit better than white light. But there was a really nice study out of Germany that showed that the white light detection of adenomas actually caught up really quickly. And there seemed to be almost a teaching effect of these new technologies that when it flags polyps that you missed before, it teaches you to recognize things that maybe you could recognize with your own eye anyway. But now that it's flagged it for you, over time, you learn to recognize them even without the technology. And so that may happen with AI, is that the more we use it, the more we may not necessarily need it. But it is a good teaching tool because it tells you what you're missing.
Dr. Wallace, Miriam, in your very recent top 10 tips in GIE on the AI, you talked about quality control. With all the different algorithms coming out, how do we control for quality? How do we know the one that we're being sold is the one we should be getting, or we should be getting a different one?
Miriam, do you want to comment on this? I know in Abu Dhabi, we had the luxury of, we had one of the systems, the CAD-eye system in a couple of rooms, and we had the GI Genius in one room, so we could sometimes go back and forth. What's been your impression doing colonoscopies with, sometimes you do them with one system, sometimes you do it with another. What's been your observation on the different systems? Are they pretty equivalent? Are they significantly different?
So I noticed that it's a bit equivalent. So far, whenever you found something on colonoscopy, a bounding box will pop up in your face, notifying that this could be a polyp, and you change the color to see what kind of polyp is it. Is it hyperplastic? Is it adenoma? So I did not see any significant difference between the different modalities, to be honest.
Do you
How about you,
embark?
Dr.?
Oh, go ahead, Dr. Wallace. Mike?
I was going to say there's been just a little bit of head-to-head comparison. For example, if you've got recorded videos, you can run those recorded videos through both systems in retrospect and compare, did they both detect it? And we're just at the very early stages of studying this, but at least the preliminary evidence looks like, as Miriam said, that they're pretty equivalent. I think all the technologies, right now we've got 3 approved technologies in the United States: the Fujifilm system, so-called CAD-eye, the Scout system from Iterative Scopes, and the GI Genius system from Medtronic. I've used 2 of those systems pretty extensively, and I agree with Miriam. They seem to be pretty equivalent to me in terms of what they're detecting. But
No,
sorry,
go ahead.
Sonia.
I'm sorry. I guess moving forward, how do we choose? How do we know what the tools we have to understand what is good or not?
In a perfect world, we would do randomized trials of those, or in this case, it's pretty easy to do head-to-head trials because you can run these videos in a kind of a simulated way, run videos through all systems. And I think that's going to start to be performed. There's not as much of a market drive and a regulatory drive, though, for those trials. The first one was obviously required 2 randomized controlled trials. That's what the FDA wanted to approve it in the United States. Everything else, as you know well, once you get one device approved in the US, everything else can come in under a so-called 510K exemption. You've got a predicate device that's approved, and all you have to show is it's equivalent to the approved device. So it's going to be hard to convince some of the companies to invest in the very rigorous randomized trials. So I think we're going to be left with some pseudo trials or simulation studies, a little bit less satisfying, but I think in the end, people will use it. And this kind of study that Miriam led, it's a really good example of a fairly efficient study design that gets you an answer pretty quickly. I mean, it's a nice case control study. She matched cases with the GI Genius against similar surveillance, diagnostic, screening exams, similar age and gender, controlling for endoscopist, and it seems to work. So those kinds of studies can be done in practice.
Should we be doing that? Should we be doing that after the implementation? So sort of internal quality control of looking if that makes a difference in our units,
Yeah,
Miriam,
as a
or
I think so. As a clinical investigator, I think we should study well, what we're doing, and know that, if one system is inferior to another, we should know that and use the systems that work well.
But that may be one of the challenges that you face when you're trying to implement AI in your practice, because most practices can't afford to bring in. Of course, you could demo something, but usually when you're demoing a product, it's for a couple days, a week at most. And it's very hard with regulations within hospitals within the US to bring in new technology. It's just very challenging. So I don't know if you could comment on the major challenges you may face when implementing this technology.
Yeah, I think, and Miriam can speak to this too. When we first implemented it, we saw both positive and negative things about it, and I've heard this anecdotally from many other groups. So when you turn the machine on, and typically we do it when we get to the cecum, because we don't necessarily want it flashing while you're washing and inserting. So we go to the cecum, we turn the machine on. It's just an on button on the front of the machine. But the first thing that people will say is, you get a lot of these false alerts. You get a bounding box around the bubble, around the stool, around the little diverticulum. And so you get a lot of these false alerts. And at first, I think people find it a little bit annoying. The reality is that those false alerts are literally lasting just a split second. There's actually a nice study out of Cesare Hassan's group that shows that the false alerts are something like 0.2 seconds in duration, but you notice them, and there's a lot of them. But you have to learn pretty quickly, which ones can you ignore and which ones are real. My own practical experience is that if it flashes a green box and then it goes away in a second, it's a false alert. If it stays on the lesion, it's almost always a true positive. It also flags tiny, tiny little polyps too. And some people, sometimes if you're trying to do a colonoscopy and it's the end of the day and you find 10 little 1 millimeter polyps, now we should be removing those, right? Except for the hyperplastics, but it can get a little bit annoying that it keeps finding all these very tiny, probably not clinically relevant polyps, but once you find it, you are kind of obliged to remove it if it looks like an adenoma.
Do you embark on any of the cut and discard techniques or processes to your practice? Now that
Not
you have
intentionally, but we all lose polyps sometimes. You snare them and they fall away. And in those cases, if I unintentionally, fail to find or suction the polyp through, or it gets disintegrated in the suction channel because it's so small, I just call what I see. I say, I think this was an adenoma. I'm going to treat it like an adenoma. But the current systems, the AI systems currently we have in the US, are not approved for the so-called CADX, the diagnostic. And as Miriam knows, in the UAE, they do have approval. So when it finds a polyp, it tells you hyperplastic or adenoma. And if that's a 2 millimeter hyperplastic in the rectus sigmoid, and you're highly confident in that, I do ignore those.
I don't know about any of you, but in one of the hospitals I used to work at, if I had a tool like this and I was in the rectum and it was identifying all these little nebulous hyperplastic polyps, and I was like, oh, it's been a long day. It's the end of the day. And there's no reason to be resecting all these. I could just see the nurses just like, oh my God, getting written up or something, or concerned that Dr. Pritzky is not removing every little polyp because it's detecting it now. I don't know.
The guidelines have given us the latitude. The guidelines now say if you have a high confidence that it's a hyperplastic polyp, less than 5 millimeters in the rectus sigmoid, you don't need to remove all of those. So I did that before we had GI Genius, and I continue to do it.
What about any other challenges that you see with AI? Like, for instance, what do you implement? What other tools do you implement to help you look behind folds?
So one of the things that I think is exciting about this space is we're quickly moving to this sort of platform type system. So right now we have a single app. And you've seen in the literature, there's an app for, although they're not yet FDA approved, but people have shown you can detect Barrett's dysplasia, you can detect UC, you can classify the Mayo score for UC, you can do bowel prep classification. All of these have been studied and reported. What one company is doing is with GI Genius is they're going to open up their platform to third-party app developers, much like your smartphone. Anybody who writes an app for a Boston Bowel Prep classifier, once they get approval, you can host the app on their technology. The first of these is coming out now, which are polyp sized. There's a polyp sizing tool. There's a withdrawal time calculator, so it sees when you're at the cecum, it sees when you're retroflexing the rectum, and it subtracts the clock. And these will start to get integrated into the report. So we're going to see 4 or 5 of these apps come out very shortly. It was just recently announced. But I think many of the things that we do through endoscopes, in the colon, the things that we have to report, Boston Bowel Prep, even things like, did you take out the polyp with a snare or a forceps? Did you retroflex in the rectum? All those things that we spend 5 minutes doing in the report, these apps will start to generate that report for you, and in the end save hopefully some time.
Miriam, any other tools that you foresee or dreams in the future that we can do, or maybe for IBD?
So I think AI is very advancing in IBD now. Like, we started to see the intestinal ultrasound that's been investigated thoroughly, and I want to go and have a course in that regards to start to implement it maybe in the future here. As Dr. Wallace said, a lot of implementation in AI will be advancing soon in regards to colonoscopies, gastroscopies, including even the bowel preparation, Boston preparation. So a lot of things will be seen soon, inshallah.
Now, I know Dr. Wen has had some interest in, we've discussed the interest of using AI for endoscopic ultrasound. And any comments on that, or helping us with cancer detection and pancreatic cysts, other pancreatic lesions, for example? Where is that headed?
Yeah, so this is actually a very active area of research for our group here at Mayo, Florida, and our partners at Northwestern University. So as Dr. Wen knows, we look at, and you as well, Mary, we get all these cysts of the pancreas, and the vast majorities are small, low-grade IPMNs or other low-grade cysts. But it causes a great deal of worry. They end up getting EUSs and MRIs and close follow-ups. One of the tools we've recently developed was an MRI-based tool that is a cyst classifier. So it takes an IPMN and it classifies it based on, we took, like all of these studies, we took people who had confirmed high-grade dysplasias or cancers based on surgical resections, and we took many cysts that were low-grade based on either surgical resection or long-term benign follow-up, and trained algorithms to classify this on MRI. And we put it up head-to-head with, we compared it to our best pancreatic radiologist here at our institution, and it was equivalent to those. Now, likely we would like it to be even better, but the reality is, your go-to radiologist when you want to say, is this IPMN something I should worry about? But they may not be reading that day. If we can train an AI algorithm to be as good as your go-to radiologist, I'm pretty happy with that, because it's probably better than the person reading it in the emergency department. The other area that's been really exciting has been the missed lesion detection. So there was a really nice study out of actually Mayo Rochester, Ajit Gunka in Gastro last year, where they took people who had pancreatic cancer diagnosis who happened to have had a scan. We see this in Tumor Board all the time. They had a scan 2 years ago, 3 years ago, and it wasn't called at that time. And they had a few hundred of these patients, and we just finished the same study here. And they went back in time and looked at that scan 3 years before their clinical diagnosis and trained algorithms to say, was there something detectable either by a human reread or by an AI reread. And they threw in a bunch of normal exams just so it was a good quality study. And about 50% of those so-called missed cancers were actually detected by these tools. So I think one of the things that we're going to see in the radiomics AI field is a lot of scans that are done in the emergency room for appendicitis or a gallbladder or something, an IVP CT for kidney stones, that they're not really looking at the pancreas, but it's there. If you build these software tools in to flag it and say, yeah, there's actually something worrisome in the pancreas, you may want to have a closer look or do a dedicated MRI or EOS. So I'm pretty excited about that area, is that we can build software tools to flag abnormalities in our case, the pancreas, but it could be a liver lesion, it could be a kidney lesion, because radiologists, you know how hard they work at reading hundreds and hundreds of images. Literally on an MRI scan, you've got hundreds of still images on 30 or 40 patients a day, and you really just can't read every one of those. It's the same problem we have in colonoscopy or in video capsule, for example. The reading fatigue becomes an issue. So I think we're seeing it across the spectrum, but radiomics to me is a very exciting area. And if we could catch a pancreatic lesion 3 years before it's clinically detected now, it gives us the opportunity to detect it at early stage, potentially shifting the survival curve of pancreas cancer.
As an expert in the field, how far away are we from implementing that in our practice?
The algorithms that we trained at, for example, Dr. Gunka's group, he was the one that published the gastro paper in 2022. So there's some regulatory issues around this, but there is actually some opportunities that you may not necessarily need full FDA approval. If you use it for internal use only, you're not selling the product. And many hospitals around the country are starting to use these tools internally as an assistant, to flag lesions. Even things like EKGs, other, routine laboratory tests. So I think it may come quicker than we expect. I don't think it's going to require, the full FDA approval, at least for internal use. Now, ultimately, if you developed a software tool that you're going to sell through your radiology or, likely your imaging companies, then that will require a more rigorous and longer process. But I think we're going to start to see lots of AI tools. Mayo's, I think, recently, announced they've got about 160 to 180 AI tools in clinical practice right now, much of which are these internal use software tools.
Yeah, this is super exciting to me and sounds definitely like the future, but this brings up ethical considerations also. And I was wondering if you can discuss any of these concerns related to the use of AI in gastroenterology, and such as issues around autonomy or over-reliance on AI technology. Any thoughts on that? I'm sure that's a big topic too that you've been a part of discussions.
Yeah, and a really important one is diversity and bias. Most of these AI tools were developed in populations, for example, in the people who come into our hospital are not as diverse as many, larger public hospitals, and certainly, larger, the rest of the world. It's one of the reasons that, I was very excited about Miriam's study is, a technology that was developed in essentially a US and European, largely Caucasian population, seems to work well in a very diverse, the United Arab Emirates is an extremely diverse melting pot of people from the Middle East, North Africa, also Europe. But I was really pleased to see that it worked in that diversity, in that diverse population. But I think we need more studies like that, where you develop an algorithm in one population, you're obliged to really test it in a variety of populations to make sure you're not just training it to detect, things that we think it's detecting polyps, but sometimes, as we know, it can detect other factors that are associated with polyps, whether it's race or gender, and it's not actually detecting the polyp itself. And back
And
actually
what
to your
other-
original question though, Mary Lee, is
Yeah,
could we become over reliant? I do
Yes.
worry that somebody could get to the cecum and do this very rapid pullback thinking, oh, the AI tool will detect it. And so if I pull back in 30 seconds and the AI didn't detect it, it must have been a clean colon or clear of polyps. At the end of the day, at least with colonoscopy, it's only as good as what you put on the screen. If you don't tip your scope behind folds, if you don't wash the colon well and distend it well, it's not going to detect these lesions. You have to be a good endoscopist first, and then let it help you become an even better endoscopist. But it's not going to make up for simple things that we still have to do day in and day out.
I have a question from the EUS standpoint. Two things in clinical practice that's difficult for us is differentiating a tumor versus an inflammatory process when you have a tumor, when you have a mass. And the second question is, when you have a mass, where do you target your biopsy site? I know there's some data, looking at both diagnostic and where to, focus the biopsy onto by EUS-FNB. So how far away are we? Will we have something similar for polyp detection in the colon in EUS application?
Yeah, thanks, Phoenix. I think there are a few studies out already on this topic that suggest it works pretty well. And there's other technologies like contrast enhanced EUS, the elastography based EUS that seem to help you distinguish if it's an inflammatory soft lesion or a hard neoplastic lesion. I would say I'm a little bit less, or I would say a little more skeptical. I would say that we're going to see AI tools in this quickly, just because it's such a niche field. I think people are going after the low-hanging fruit first, the things that we do hundreds of thousands of times, if not millions of times a year, like colonoscopies, maybe screening for Barrett's. I think although you have a passion and expertise for EUS and the pancreas like I do, and like others do, I think it's still, if you're a company developing a product, you're going to go after where the market is first. So I think in these areas, I think it is an opportunity to develop some of these in-house uses. And it looks like the FDA will likely allow if you're using it for internal purposes only. But I think it's going to be a longer road for large commercial products in these niche fields.
Any other questions? Dr. Riker, Phoenix. So I guess two things is one we talked about earlier before we started this is, what do you see the future for the broader scope of the practice of medicine? How is it going to help us with our electronic medical records? I find it very cumbersome throughout the day if I have a complicated consult and I'm digging in a chart. And yes, I'm lucky not to have huge paper charts in front of me, but I'm looking under labs, I'm looking under media, I'm looking under pathology, like I'm trying to put everything together. Can AI sort through this? If the patient comes in with a specific complaint, can it start pulling everything in a comprehensive fashion for me?
Yeah, I think this is an area where it's going to be, I think, quite helpful. So, I mean, to take your example, somebody who comes in for a tertiary hospital, second, third, fourth opinion, traditionally they brought a giant stack of records. Now there's probably hundreds of pages of PDF files that probably got uploaded onto your electronic health record. And, take an IBD patient that Miriam sees, and maybe they've been treated with 3 different biologics and, steroids of various forms and other disease-modifying agents. And you're just trying to figure out what have they been on. And you've got 100 pages of a PDF file, and you want to know, did they already get Stelara? Did they already get infliximab? Well, one of the really nice things about these large language models is you can query them. So once you upload that PDF file, you can query it and say, did this patient ever get Stelara? Did this patient ever have a drug reaction to infliximab, and it will search for those words. And so I think it becomes a searchable document in a way where, we've all used ChatGPT a little bit. You can search for things. It will have to be within a HIPAA compliant environment. So that's really critical. You don't want to upload a patient's PDF files onto the ChatGPT public site, because that's a complete violation of HIPAA rules. But many hospitals are developing in-house or ChatGPT equivalents within your firewall. So I think that'll really help us a lot. Another example is back to my sort of area of interest in pancreas cancer. You all, I'm sure you've all seen this literature that one of the earliest indicators of pancreas cancer is a slight rise in the blood glucose and a slight drop in your weight. But most people don't pay attention to that. They say, oh, the glucose was up 5 milligrams per deciliter and their weight was down 3 pounds this year. But you can train algorithms, and this is one of the projects we're working on, to detect these really subtle things. Even low, if your cholesterol is dropping a little bit, that's actually a pretty good predictor. If someone's weight's dropping, their cholesterol's dropping, and their blood sugar is going up, the so-called NPAC score is a pretty good predictor of pancreatic cancer risk. But you need a search tool running in the background of your EHR to pick that up, because we as clinicians aren't looking at the subtleties of that within the EHR. So I'm actually quite excited about these more sort of mundane things of searching for subtle, clues within the text base using large language models, and other clues looking within, for example, the laboratory records for subtle changes in labs that might indicate a disease process.
How do you use ChatGPT? In one of your article, somewhere I read, you said that you used ChatGPT to help you draft the manuscript. How do you guys, if you use it, how do you use it?
Yeah, there's a lot of ways you can use it. You can, and journals are pretty particular about this right now. So GIE, for example, we don't, and I talked to Doug Adler, the current editor, about this. In fact, when he asked me to write how do you implement AI into your practice, I asked him, you want me to write this from scratch myself, or do you want me to, have ChatGPT start and then I'll edit it? And he was very clear. He said, I want you to write it from scratch. I did it both ways, actually. And the first way with, ChatGPT was pretty good. I do think that common documents that we write, if you need a research protocol and you just want to have all the common verbiage that goes along with that, ChatGPT is really good at that kind of stuff. If you need a standard SOP, a standard of practice document for, how do we process our biopsy samples, our FNA samples, and we're sharing that amongst our cytopathology colleagues, ChatGPT is really good at it. And that's where, the paralegals are getting a little bit nervous. But in the end of the day, there are some things that do require human intelligence that AI is not good enough at. And that's, writing a manuscript and understanding the subtleties of what's the current literature, what are the knowledge gaps, how does this study fill that knowledge gap, how do you compare our study results to another study that may have conflicting results, that still requires human intelligence.
Oh, okay.
Are you disappointed?
No, I just thought it's going to do all my work for me, but it's not going to happen.
Not yet.
Not yet.
So ready to wrap up, I guess, speaking of artificial intelligence and ChatGPT and implementation within the colonoscope, it brings up a lot of dreams. And I think about with you, Miriam, and you're entering this growing field of IBD, if you had a magic keyboard and you could ask it one question that you wanted AI to provide you, what would that be?
What is the best treatment for this patient? Because these patients have been trying a lot of biologics, been going through a lot of rough time. So I wish if I can be provided with one answer to help them for the rest of their life.
I like it. And Mike, for you, with all the areas of passion and interest you've developed over the years, if you could ask that keyboard one question, what would it be that AI could help you with?
For me, it's getting rid of the mundane tasks that are low value that unfortunately we as physicians have to do a lot of. And as we know, the biggest issue of physician burnout right now is we're spending hours and hours a day typing reports, filing appeals to insurance companies. I want a tool that, you know, in the area now that's emerging is this so-called ambient clinical intelligence. It listens to you talking to your patient. It drafts the note for you. It may place the orders for you that you have to approve. But if I can spend my time just talking to the patient and listening to the patient and understanding their needs and focus on what we as humans do really, really well, being empathetic, being understanding of them, and let an AI technology do all the other things that we as humans, you know, either don't like to do or don't necessarily do so well. You know, write the note for me. Obviously with approval, but certainly filing insurance appeals, you know, all of us hate that. So that's my wish.
Well, I'd like to thank both of you on the behalf of AWE for participating in this Q&A and also participating in our conference this year. And hope to see you again soon. Thank you so much.
Thank you so much.
Thank you
Thank
for
you.
being here.
Yeah.
Thank you so much.
Thank you.